Hi everyone, it's Mrs. Bustamante. Today we're going to talk about 2.4, which has to do with the plasma membrane. Our main learning goals are going to be able to describe the role of the components that make up the cell membrane and their importance in their environment and their importance to the cell, and then to just describe the fluid mosaic model of the cell membrane and talk about how it's put together and why it functions the way it does. All right, let's go. So the first thing is, what is the function of the cell membrane? Well, it does a couple of things. Maybe the most obvious is that it provides a boundary, right? The cell membrane's goal is to maintain an internal environment. And to maintain an internal environment, we have to have some sort of boundary, and that is the cell membrane. It also helps control what enters and leaves the cell. Sometimes we talk about it in regards to transport, just moving things in and out. And when we talk about the cell membrane, we're talking about it being mostly made of phospholipids. And this is a basic phospholipid here. We have a polar head, it's the phosphate part, and then we have two nonpolar tails. And those kind of combine together to make what we call our phospholipid bilayer. So let's discuss a little bit more what a phospholipid is. So a phospholipid has what we call a hydrophilic head. It has a phosphate group on the end of it that makes it polar, and then it consists of two hydrophobic tails or fatty acids. Here, one you see is a saturated fatty acid, the other is an unsaturated fatty acid. But we have two components, right? This part, our fatty acid tails, are going to be hydrophobic. They're not going to love that water. And our hydrophilic head is going to like water. So because they make up the majority of the cell membrane, they play a really important and key role in maintaining that structure and the function of the cell membrane. And because they have this hydrophilic portion and this hydrophobic portion, we call them amphipathic. That just means kind of they have both parts. So what tends to happen? Well, the hydrophilic heads or the phospholipid head will go towards the watery environment. This is outside of the cell and inside of the cell towards the cytoplasm. And this hydrophobic or fatty acid tail kind of ends up in the middle. So what do you get looking like? Here's your phospholipid bilayer. They sort of line up. We call them 2 bi together. Matching up, this is your lipid bilayer. And this will happen spontaneously when these phospholipids are put into some sort of aqueous environment, because this hydrophobic portion is going to want to get away from that water-like substance, and the polar portion will want to stay towards it. In our phospholipid bilayer, there are also these things called proteins, and proteins can be embedded in our phospholipid bilayer in a couple of different ways. We can have hydrophobic proteins, hydrophilic proteins, or even both, the combination of the two. Peripheral proteins are going to be the ones that are just kind of on the outside. They tend to be more loosely bound and are kind of, you can think of as connected to the phosphate part. Then we have integral proteins that go through your entire phospholipid bilayer. So they have to have parts that are hydrophobic and parts that are hydrophilic. They span the membrane, if you will. So these peripheral proteins are typically hydrophilic because they interact with the charge or the polar sides of the group, and they're on the outside or the inside, right, of your phospholipid bilayer, which we know is aqueous. Integral proteins have to have a hydrophobic portion that interacts with this lipid tail because they go all the way through our phospholipid bilayer. And they have to have a hydrophilic, portion that interacts with the polar region of our bilayer. An example of these are transmembrane proteins, and we call transmembrane proteins that basically go all the way through the phospholipid bilayer. Sometimes they can also be channel proteins that we'll talk about later on too. So what is the function of these plasma membrane proteins? Well, they actually have a lot of different functions, and we're going to talk more about them in later units. But they can provide a lot of ways to allow things to come in and out of the cell, or rather transport is one of the main ways we talk about them. So they can be selective through a channel protein, maybe only a certain part or particle will fit through. It's really a mechanism to only allow certain things in and out. They can have enzymatic activity, meaning they can trigger certain events, right? They can provide signal transduction, which means it's going to send one signal onto another place, onto another place, onto another place. And they can help with cell-to-cell recognition. If you can see in this picture, these are little hexagons. And if we think back to our unit on macromolecules, we can think about hexagons as being sugars, right? So these are typically carbohydrate type chains that can help identify the cell to other cells so they can interact appropriately and sometimes join with other cells. It can have intracellular joining. And then it can also provide kind of like attachment or anchor points for the cytoskeleton to help give the cell its strength and its rigidity that it might need. So what are some of the other components of the cell membrane? Well, another major component is this idea of the fluid mosaic model when we talk about cell membrane. So what does that mean? Well, fluid means that the cell membrane is actually mobile a little bit. It moves. It's not static or still or stuck in one place, if you will. And it's held together by hydrophobic interactions, which if we think back are weak interactions. They're not going to be very strong, which kind of allows for this movement. Lipids and proteins, therefore, can shift and move on the surface of the membrane and across the lipid bilayer because it is fluid, it is movable. Some other components, cholesterol is really important in the lipid bilayer. It's a steroid and it can be randomly distributed. Here in this picture, it's kind of these lime green areas. And it's typically sandwiched in between the membrane, particularly in eukaryotic cells. And cholesterol is really important because it helps control fluidity and flexibility in the cell. So sometimes at really cold temperatures, the plasma membrane will sort of slow down, not be able to move as much. But what cholesterol can do is it sort of helps it stay flexible and not freeze, so to speak, if you will. The other thing it can do is if the plasma membrane gets really hot, it might kind of start to melt. Think about it melting. And cholesterol helps negate that too, helps it stay kind of fluid, but not melty fluid. The last component we need to talk about are glycolipids and glycoproteins. And they're really just carbohydrate portions that function in cell regulation typically. Glycoproteins are one or more carbohydrates. They're attached to the membrane, so you can see it here. Here's your glycoprotein, and it's attached to a membrane protein, this little blue part here. And glycolipids are connected directly to the phospholipid, the lipid part or the lipid group in the phospholipid bilayer. So they're more of a direct connection. I hope that was helpful today. I hope you understand what the phospholipid bilayer is and the importance of the plasma membrane.